Thank you. I'm Kelly Rowland. I'm one of the organizers for WiDS Berkeley. I work at the National Energy Research Scientific Computing Center up at Lawrence Berkeley National Lab. And I am very pleased to introduce our next speaker, Kjiersten Fagnan, who is the Chief Informatics Officer at the U.S. Department of Energy Joint Genome Institute at Lawrence Berkeley National Lab. Today she'll be talking to us about the National Microbiome Data Collaborative. Take it away, Kjiersten.
Thanks, Kelly. So yeah, I'm happy to be here. I was glad that Kelly invited me. Actually, for the last year and a half, Kelly has also been working with the Joint Genome Institute and helping transition some of the bioinformatics type workloads over to supercomputing environments, which is kind of a different beast altogether. So after Danny's talk, I wanted to maybe pull the audience a little bit on different data types. So how many people here know what an image looks like? Yeah, all your hands should be up. Yes, that is accurate. Okay, good. If I were to transmit to you a JPEG or a PNG, raise your hand if you know how to open that. All right. See, look, we're feeling good. We know how to deal with certain amounts of data. And also, you understand that when you're given an image file, it's a picture of something. It's a digital representation of some sort of graphic, something you should be able to interpret. Most people around the world, like you can give them pictures of things, and Google got really smart about this, right? Like the CAPTCHAs. We got really good at labeling their data for them, right? Because that's just something that's easy to interpret. We all know what stoplights look like, cars look like, mountains look like. We might not know what cancerous cells look like. So there you're kind of delving into the realm where you need some scientific expertise. But in general, you can kind of deal with images. And I think that that's really where a lot of progress has been made in industry. I'm not trying to be reductive, but like image processing has been really important for a lot of super cool and interesting applications. But when you move and you pivot into scientific data, it gets a little weirder. So how many people here are familiar with the concept of a genome? Excellent. Probably even microbiomes, yeah? We've all heard about probiotics and how awesome they are for us, particularly with an emphasis on women's health sometimes. But how many people here have seen a FASTA file? Seen a sequence data file? That is more than I would thought. That's great. How many people here know what to do with one of those files if they're not told where it came from? That's where I kind of expect the hands to not go down. Without the information, like I can give you a string of A, C, T, and G, right? And you might look at that and say, cool, all right. You know, like you don't necessarily know what to do with it without some sort of contextual information. Without me telling you where the sample was taken from and what you might expect to see in the sample, you can't tell if the genome you're looking at comes from yourself, comes from a microbe, comes from a plant. Like there are signatures you can look for if you're a biologist and you know what you're looking for, but we can't just give those data sets to everyone and have them help us sort of classify and figure out what's going on. And then it gets even weirder when you start looking at RNA or when you start looking at proteomes, which is, you know, the protein signatures of what's being generated in a system, or metabolomes, you know, secondary metabolites and things that you're actually, or the microbes that you might be studying are actually creating to try and understand what's going on in a system. So we've all heard of the human genome, we've all heard of genomics, and that's created this kind of really interesting space for advancing our understanding of biology. And I think as computational folks, and it looks like we've got some bioinformaticians in the audience, you know, we start to think about that as a blueprint. It's like really interesting to think about how to use that and think of genes and think of some of that information as switches you can turn off or on, and that might do something in a biological system. But it gets really complicated. We've had the human genome for a while, and it's not like we're necessarily hacking one another's genomes anytime soon and really turning things off and on because it's really risky. Even in a microbe, which are these small genomes, you can try to turn things off and on, but then they basically self-destruct. They're like, you know, I don't actually want to do that. So if you want to take a microbe and get it to produce something cool like a biofuel, or some of them, and for those of you who have heard me speak before, have probably heard me say this because I think it's so cool, some microbes actually excrete magnets. Why? What are they doing? What is going on? Like, it's so cool. And pretty much for any kind of like weird thing we can measure in the environment, there's probably a microbe or something you can't see, a microorganism that's involved in that. And so what we're trying to do is really understand what all of these critters are doing so that we can kind of take them and use them to produce things like, there's a very cool project where they actually hacked the genomes of microbes so that we could create more insulin in the laboratory. That's an amazing application. They're also hacking the genomes for mosquitoes and making it the case that the mosquitoes that are carrying malaria or prone to carrying malaria, which are the female mosquitoes, actually can't reproduce. They're sterile. So there are experiments like that, and there's clearly a lot of like interesting ethical conversations you can have related to that too, and unleashing things in the wild. But these are things we're still fundamentally trying to understand, and the objects we're trying to understand are living organisms. How many people here have tried to get another person to do something you wanted them to do that they weren't sure that they wanted to do? All the time, every day, that is life, right? And so now imagine that you can't necessarily easily communicate with that other organism, right? And you're like, I'm gonna poke you, and then I expect you will excrete a magnet. And it's like, no, that's probably not what they're gonna do, right? And so we're still in this space of trying to understand it and trying to catalog all these creatures, and then trying to think about how we can convince them to work with us to do some of the things that we want, how we can convince them that it's in their best interest to make insulin, how we can convince them that it's in their best interest to make chemicals that are precursors to all of the materials that we need in our awesome clothes, therefore we can remove our dependence on fossil fuels. So fossil fuels are used, everything that we're wearing has fossil fuels as a precursor. It's a little unfortunate, but that's our reality. And so what we'd like to do is actually hack some bacteria to develop different chemical precursors that don't depend on that. So there's this whole wide space of trying to influence all of these little organisms that we can't see that actually drive the health of our environment, drive the health of our bodies. I know there are things that we typically get scared of, like in the bathroom. You're like, oh, germs, right? Viruses, coronavirus, scary. Right? So like all these microorganisms you typically think as being kind of bad actors, but there's so many more of them that do awesome things. And I am a mathematician, so I started working at the JGI not that long ago, and this is all that I have really learned or come to appreciate, is that all these unseen objects are actually kind of driving and dictating existence. And this doesn't mean that you can sort of abdicate all responsibility to the microbes and the microbes made you do it, but like they really do kind of drive a lot of what keeps our planet healthy, which I think is pretty cool. Danny did a really nice job introducing the national lab, so I'm not going to belabor that, but this is a completely different effort, the National Microbiome Data Collaborative, which is geared toward collecting information about all of these microbial communities where we've collected samples and tried to sequence them in one place with all the contextual information that we might need to understand, if I were to give you a FASTA file, that it's actually like a set of organisms and microbes that you might be able to identify, like E. coli. And you'll have to forgive me, my knowledge of nomenclature for all the different microbes is not that great. So I can't give you the name of the cool example, but I can tell you things like they excrete magnets. It's cool. I mean, it's a skill. So one of the things that I wanted to kind of share with this audience is omics data, which we've kind of talked about a little bit. It's more than the genome, and it involves things like the transcriptome and the proteome and metabolome. This is like the central dogma of biology that we're still trying to understand, but we believe and understand this is what's happening in your cells every day as they're actually like producing the proteins and things that you need to live. To actually be able to do this across all taxonomic scales, ecological scales, and even these genetic scales, it's just an enormous amount of data and requires an enormous amount of computing. But because we don't have this kind of, and I'm not going to say that it's simple, except maybe in the context of this it is, like we don't have images we can just share with everybody and have you all help us label all of these data. We don't have, I think it all gets very complicated and very messy because that's what I was trying to say earlier. Biology is just messy. These are living creatures that have a will of their own. Really, it's a will to survive. And like you're trying to catalog and understand that. If we were to actually write down actually just the differences in the genomes of all microorganisms, how much storage do you think would be needed for that? So you've got maybe a terabyte on your laptop. Maybe, like, Danny was talking about, we're now generating terabytes a minute. Eventually, it'll probably be on the order of tens, hundreds, and then petabytes of data a minute. To actually just write down the genome of all microbes would require a yottabyte of storage. That's more than we've got right now, just to be clear. And that's also kind of weird because, okay, how many people here have heard of strains of the flu? Like you go get a flu shot and you're like, oh, I got the flu anyway. And it's because you got a different strain that your body hadn't seen before. That's also really weird and hard because if you were actually to capture all of the different strains for E. coli and you expand and blow up the footprint of what it is you're trying to catalog, then you need many, many yottabytes to actually store all of that information. And that's just microbes. That doesn't even include us. I know we think we're really cool, but our genomes are actually relatively simple compared to plants. I'm just throwing a bunch of what I think are cool bio fun facts at you guys, but just this curiosity and appreciation that you can develop for these other fields is what helps you get interested in the mess that is biological data and the scale that is biological data. To actually be able to go through and deal with this volume of information, we do need humans to look at it. We need people with expertise. We need to get an understanding of what those humans are seeing when they look at the data so that maybe we have some hope at doing a better job actually labeling and cataloging all of this information. But from where I sit, it feels insurmountable most days. So what is the point of the NMDC, which really I should get to because I realize that also we don't have a huge amount of time. Oh yeah, I love this drawing. It's sort of, because it gets worse than just the scale. Also, if you think about it, you've got people generating data in individual labs because they've got a hypothesis that they want to test. They're tracking everything, they're doing the best they can, they've got lab notebooks. Maybe if you're lucky, the code's in a Jupyter notebook, or maybe they're using version control, but generally speaking, there's all kinds of stuff happening in the wet lab that needs to be captured for the data to be useful to you and me, right? And just think about that mentality. You're doing an experiment, you're answering a question, you've gotten funded to do it, to actually make those data useful to anybody else who's not in that is hard. It's hard to think about what they might need ahead of time. And so there are organizations that have cropped up to try and help think about what those data standards can look like to actually make it easier to share information, to make it easier for me to do an experiment and then pass it off to you and say, what would you like to do with these data? But it's not easy. And it definitely looks like this. I can say after 8 months of this project, we're taking data from 2 national user facilities. So NERSC, which Kelly mentioned earlier, is sort of the backbone for supercomputing. But we also have a user facility in genomics and one in a whole bunch of other measurements like proteomics and metabolomics. And we work together. We had joint projects, but they were all for a single PI. And now we're trying to take all of the data from those joint projects and figure out if somebody sent us a sample and it went to EMSL, the Environmental Molecular Sciences Laboratory, if they actually took subsamples and sent us the same sample at the Joint Genome Institute. So we're trying to figure out if the metabolome and the proteome that were measured from this sample A actually have the same sequence information associated with it. And the only person who knows is the PI that wrote the proposal to both organizations, because we're looking through metadata, we're looking through lab notebooks, and we can't figure it out. Because no one thought in the moment about wanting to use these data beyond the PI who came up with the proposal. And you might look at it and be like, wow, that's really silly that happened, but this happens all the time because we're moving quickly. There's a lot to do. So it's kind of a fascinating learning experience to kind of go back in time and think, well, how could we do this better moving forward? Because fixing all the historical projects is really just maybe not going to happen. So yeah, we would like to get to this kind of beautiful, organized world. It's a dream. And I will say that this project has this kind of interesting background. So one of the things that's kind of cool about working with microbes and microbiomes is that it goes beyond the Department of Energy. The Department of Energy has just been the first to step up and say, hey, we're going to fund this kind of data curation and organization effort because we know that it's needed to advance our science. We know that it's going to be needed to actually approach understanding the data at the scales on that picture I showed you previously. But this is of interest to the NIH, USDA, EPA. It turns out everyone is studying microbes. See previous statement on them driving the world around us. We want to understand them. We want to understand what these communities are doing. We want to understand how this influences the health of crops, the health of humans, the health of babies when they're being born. And that requires having an enormous amount of information that's well curated and well understood so that you can do a data analysis experiment across all of these different samples. And so this is being funded by DOE at this moment, but we are kind of hoping that in the long run, this project will actually spin out into a life of its own. In an ideal world, the National Microbiome Data Collaborative becomes a data resource that's run and managed by a nonprofit that could accept funds from all of these different agencies that can kind of be there as this kind of long-standing resource for the community that isn't necessarily owned by DOE or EPA or the government. And so figuring out how to make that happen is going to be a whole different challenge. So I might just end with this kind of vision for what it is, because I think that this slide kind of captures a lot of the activities, and I would be happy to answer questions or talk about this at lunch. But we want to empower the research community. We want to empower not just biologists, but also computer scientists, those that are developing algorithms, statisticians, mathematicians, to help us get a handle on these data. And we think that the only way to do that is to create what is effectively going to be a very powerful data catalog type resource with a very powerful type of search so that someone can come on to the NMDC, think of it like a data catalog, and they can figure out, okay, I'm interested in this particular organism. I want to be able to search by that organism's name, and I would like to see all of the data sets that contain this organism. And I'd like to see information about the quality of those data sets. I'd like to understand the geographic location where these data sets were taken. I'd like to understand the protocols that were used in the lab. It's a lot of this contextual information that's going to let a scientist figure out if a data set is of use to them. So to do that, we think that it's really important to have clear data standards around the metadata and also the quality of the data. That we're going to reprocess all of the raw data with the same pipelines, the same sort of quality control pipelines, the same annotation pipelines. So when you're annotating something, you're creating that context. You're basically saying, okay, here's an unknown string, and now I'm going to do a lookup and see if anyone else has seen this string before, and then I'm going to assign those characteristics to it. It's very basic and not always completely accurate, because you can imagine if those databases of things you've seen have names that change. So that's the other cool thing about biology, because we're still cataloging a lot of things, the names change all the time, all the time. You're like, oh yeah, I thought that was an E. coli, and it turns out that it's yeast. It's not that dramatic, but like those kinds of naming and nomenclature changes make it really hard to go back to a static publication. So you need these kinds of, like, for lack of a better way to put it, like living dynamic resources that can allow for the nomenclatures to change. And then if you don't process everything in kind of the same way, it becomes very hard to know what data could be integrated. And data integration is a term that really bugs me. I don't know if other people in the room feel the same way. But as a mathematician, I'm like, are we talking area under the curve? Because I think that's the first time I ever saw that, right? Or are you talking about bringing in these like disparate data from different locations and then trying to push them together? And what does that mean? What's the underlying scientific motivation for needing to integrate data? And so in our case, the multi-omics analysis, being able to integrate the genome and the transcriptome and the proteome, like that's all driven by a desire to have better biological understanding and things that have been observed in the laboratory. That might mean that you want to validate this whole flow of information with an image of a protein structure, if that's something you understand. But you also might want to validate by the fact that you modified a microbe's genome. Did it die? Did it survive? Is it producing what you thought it should? Is it producing the magnets? I would really love to do that experiment. I have no qualifications to do said experiment, but I at least want to like see it happen, just because it's so cool. And then we want to make sure that we can use this to facilitate access to the data. If you would like 10,000 metagenomic data sets to do question mark with, so you have enough for some sort of statistical analysis, we want to make sure that you actually can do that. You can access the things that are public. You can get it at the level of interest to you because there are going to be different levels of processing. And we think that to do that and to enable that kind of search, we have to process everything kind of uniformly. We have to have the same context and metadata around it. And then we can let you search by gene. We can let you search by organism. We can let you search by environment, search by pH. But we can only do that if the scientists actually provide us the pH information and the protocol information. And so part of this is a feedback loop. Remember, getting humans to do things that maybe they don't want to do. Like showing that there's value in building a system like this and that it actually amplifies their research motivates people to do kind of the boring thing of like tagging all of their samples appropriately. And the more of those that we have that are tagged appropriately, the more we can think about automatic assignment of metadata and automatic curation. But I would say we are so far from getting there. So if somebody finds that interesting, come talk to me. Anyway, I will leave the rest of the slides. You can take a look at them later. I had a lot more super fun detail and also a picture of somebody trapped in some cables. So reasons to look at the slides. But I'll take a couple of questions if there are any, or move to the next speaker. Sure.
Thank you. That was a fun talk. I was just curious, I mean, this may be at some point like way in the future, but in terms of sort of these data storage problems and sort of, you know, gathering genomic data and, you know, RNAs and all this stuff in different stimulus contexts and over time, I mean, it's going to be a huge, massive amount of data, and you were saying we don't even have enough storage space right now to store genomes, like reference genomes. So should we go more for like streaming models of data analysis where we don't have to store it all and we can just do it in real time? Or, I mean, is storage going to get small enough that we'll be able to do it when we come to that point?
How many people have heard of storing information in DNA? Yeah, that's super weird, right? Okay, anyway, to make that effective, you actually have to get really good at synthesis and sequencing, and maybe we'll get there. Like right now, it's a write-only database, so that's less helpful. But I mean, to your point, so if we were to sequence all microorganisms that we think exist, it would be a yottabyte of storage. We have not gotten that close, even close to those numbers, because it turns out actually they're kind of tricky. So you can pull things out of the environment, and then you can't actually culture them. They just die because they want to be in the environment with their buddies. And so there's a whole host of microbial organisms that we can't even collect the genomes on yet. That's really a fundamental challenge in the field. But say that we were, and we wanted to store it, or how we're dealing with storage now. We actually have really kind of amazing storage resources within the Department of Energy. We use tape. We use high performance tape, right? You didn't think that that could exist. And for those who are maybe, you know, didn't grow up in the 80s or before and have no idea what tape is, you know, it's on a spool and you read it. But yeah, I mean, we actually use that to deal with the fact that JGI, the Joint Genome Institute alone, has 13 petabytes of data, but people don't want it all at the same time. And so using this kind of hierarchical, like tiered storage model, you can kind of get around that. For the metadata and the contextual information, that actually doesn't take up that much space, or not as much space as the raw data. But one of the things that we are thinking about is, do we need to make multiple copies of the raw data or even of the processed data? Like what are scientists actually needing from us to do their work. And I think that there's a lot of information where if you capture the annotation type information properly, there's a lot that you can get just out of the metadata alone to help you understand what's there. And then it's figuring out those next steps of what people might want to do and how they might want to aggregate a whole bunch of this data together that I think requires a supercomputer in some cases because you've just got so much of it to process, but you really want to think hard about the type of question that you think you can answer with that. So there's a roundabout way of saying, like, the problem's kind of under control now. I don't know what's going to happen. We certainly have too much data to actually store in the commercial cloud and pay for it on a regular basis, so then that brings up these data accessibility issues. And what we're thinking right now is that those who are generating the data will put it into repositories like at the National Institutes of Health. The Joint Genome Institute runs our own, and so we kind of want to have these distributed repositories and then have kind of a catalog interface on top of it where you can find what you're looking for, and then we send you to that location to actually download the data if that's what you want to do. And so it's really going to be this kind of distributed data type model, and then figuring out the types of compute or questions where distributed is not okay, I think will be an interesting research problem. I have an argument with a guy who contends that like there's no problem that requires that the data all be co-located. And then if you really get into it with him, he's like, well, yeah, you know, if time isn't a factor. It's like, so when do we make time a factor? If it's going to take 10,000 years to solve this distributed problem, like maybe I don't want to wait. And so, you know, it's just, and so that's the balance. But anyway, sorry, that was a long-winded answer. Are there any other questions? Now, how do we all feel about microbes? They're pretty cool, right? All right. That's really all I wanted you to take away from this talk. And if you do have an interest in data science and algorithm development with applications to science and not to advertising, though I guess automated cars are pretty cool. But, you know, we have a lot of opportunities up at Berkeley Lab, and I think that there's a huge opportunity to have an influence and make a difference there, particularly if you're curious about this multidisciplinary type work. And it will challenge you if you're going to do multidisciplinary type work. It's really important to be curious. It's really important to like listen and ask questions because you don't know what you're going to learn. And also, like, I think, and I'll just say this, I honestly believe that women are better at this. So with that.